Hi everyone. Am I audible? I am not able to find your chats anyway. Yes, I am audible. Okay, so let's see. Chat is not visible. Maybe I'll ask them. All right, anyway, so ace your term 2 exams with Unacademy Plus. Okay, so Unacademy Plus subscription at flat 10% off. So this you will be getting, and this is with this particular code Vani YT. My name is Vani and YT is for YouTube. Okay, so you can easily remember this code. So this is basically by using this code you will be getting 10% discount on subscription, that is on Plus. Okay, so that is basically Unacademy Plus, where the classes will be done in a regular manner. So before I continue, just a second. Yep. Okay, so let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Okay, so let's start today's topic. So let's see what we are going to study today. It is the structure of kidney tubule and its functions. Okay, so we will see the structure of kidney tubule and its functions. Okay, so this is my code again, Vani YT, for 10% discount. And also you can follow Unacademy Champions on Telegram. Okay, so link you will find in description box. So subscribers on YouTube now, let's target about 1000 subscribers. You can click on bell icon. Okay, so where you'll be getting the notification whenever the classes are there. Okay, so starting from this one, this is the internal structure of kidney. Okay, so last class I was saying that we will discuss like what exactly is the cortex region and medullary region, and after that so today's session we will completely see the structure of this particular kidney tubule. Okay, so now as you can see here, they have given a longitudinal section. What is longitudinal section? So let's say this is a kidney now. Okay, so if I cut it like this, okay, if I make a cut like this, then it is transverse section. So now this is a kidney, they have made a cut like this. Okay, so cut is made like this, so a longitudinal section. Okay, so it is basically a longitudinal section where you will find two main regions. Okay, so there is a outer dark, so this one is outer dark cortex region you will find, and then there is inner lighter medulla. And when you are drawing kidney, so make sure that you are again drawing the two coatings. So here this one, so let's say you are drawing a kidney as such. Okay, so here you will anyway draw this pelvis. Okay, so there is pelvis part over here, so you know like this you'll randomly you'll draw this thing. Okay, so this one second, yeah, so this one is actually the ureter which you are joining. Okay, so now this covering, what is there for kidney, you need to draw this double covering. Okay, so there has to be always a double covering, so because there is a protective covering which is there. Okay, so other than that you will be drawing the medullary region. So this one is now a cortex region, and then there is a medulla region. Okay, so here at junctions you will find, so this striations what you find here. Okay, so you'll find these certain striations. Okay, so this striations, or what we call as a kidney pyramids. Okay, or a pyramids, what are there? So these pyramids you will find in the medullary region. So this is now all pyramids over here. Okay, so this particular pyramid when we mention, so this is actually made up of so there is okay, I'll explain about that. So this dotted appearance is there. So first let me just label and I'll explain you why exactly we have got this appearance in the kidney. Okay, so this part what you find while I am drawing this dotted region, so this is actually the cortex region. Okay, so this is cortex region. All right, so in cortex region what do you find? So basically in the cortex region, so maybe I can just write it over here. So it is the Bowman's capsule. Okay, so there is Bowman's capsule or Malpighian capsule what we mentioned. So with that you will find the PCT also. So this is will have the glomerulus as well. Okay, so basically Bowman's capsule and PCT is giving this particular appearance. So it can be DCT also. So all these we can find in the cortex region, and here what you find, so this is for cortex region. Okay, now this in the center what you find here, so this is actually the medullary region. So this is medulla. Okay, so at medulla what do you find? So at medulla you find, or you will find the structure of, maybe I can write it over here itself. Okay, so it is Henley's Loop. Okay, so there is Henley's Loop, and also the collecting duct. Okay, so collecting duct is basically the urine collection is done, right? So that you will find in the medullary region. So that is why this part what you find, so this pyramids what you find here. Okay, so these pyramid or striations or striated structures what you find, so this is nothing but the Henley's Loop. Okay, so this will be made up of Henley's Loop. Okay, and also the collecting duct will be there. So that is why we are getting this line like appearance. Okay, so this one is collecting duct, and here this one, the dotted appearance, what is that? So that is because of the region or the cortex region which is having the Bowman's capsule and the PCT. DCT also can be there. Okay, so longitudinal section of the kidney shows two main regions. So there is cortex region and then there is a medullary region. So medulla is composed of a finely striped substance arranged in several conical pyramids. So you will find many conical pyramids here. Okay, so now this is one conical pyramid, then there is again a conical pyramid, here again a conical pyramid. So this is one conical pyramid. So all these conical pyramids, what is them? So the apex of this particular conical pyramid, so now here this is the apex. Okay, so this apex of the conical pyramid, what is that? So this we call as a papilla. Okay, so this one is a papilla now. Okay, so this papilla is gonna project where? It is projecting, you can see it is projecting into where? It is projecting into the pelvis. Okay, so pelvis is there, so that is pelvis of the kidney. So next to that, so the kidney is having many enormous number of minute tubules. So they can ask you the, like in board, so they can give the name of this particular nephron as a uriniferous tubule, or also they can give it as a nephron, or they can give it as a renal tubule, or they can also give it as a kidney tubule. Okay, so remember, just remember all these names what are there, so they are one and the same. So they might ask you to draw a structure of a renal tubule. So just don't keep on thinking that what is renal tubule or a kidney tubule or what is uriniferous tubule. So it is all one and the same. It is basically the nephron. Okay, so you need to draw the structure of nephron. Okay, so they can ask you, since subjective type it is. Okay, so you can draw the nephrons as well, or they can give you a diagram based question also in this. So you should be able to solve this one. So we will see the structure today. Okay, so the structural as well as functional unit of kidney. Okay, so why do we call it as a structural and functional unit? Because you can see this kidney is completely made up of nephron only. Okay, so there are no other cells. So basically nephron, what is there? So nephron, it will be made up of a nephron. That means, so here and also this complete arrangement in the cortex region and in the medulla region, so it is basically of a nephron itself. Okay, so there you will find this nephron cells, Okay, so these nephrons are again made up of many different cells will be there, but basically this is now a kidney which will be made up of this functional unit. And why do we, okay, so this was related to structural unit, and why do we call it as a functional unit? That is mainly because it is going to perform a function where the urine formation occurs. Okay, so that is why we call it as a functional unit of a kidney as well. Okay, so nephron, when we mention, it is basically structural and functional unit of a kidney. Okay, and again it will have many different cells, it will be made up of different cells, so that you need not study in this class. Okay, so basically I'll be just explaining you about the structure of this nephron. Any doubts you have till here? I can't even see your chat. No, it is not even seen. Your chats are not seen. Anyway. Okay, so yeah, so now this is related to the urinary structure. So basically the uriniferous tubule, the nephron, renal, or a kidney tubule. And you know, so there are about how many nephrons in each kidney? So if we are talking only about one kidney, okay, so this is one kidney now, so we can find up to 1 millions of these nephron. Okay, so 1 million nephron per kidney we can find, so it can be up to those many numbers. Okay, so many nephrons will be there in your kidney. Okay, so it is 1 million, so approximately we can consider 1 million. So if two kidneys are there, then two of them together, it will be 2 millions of okay, I cannot write this side, no, you can't see this one. Okay, so it is basically 1 million. Okay, so 1 million nephrons per kidney. Okay, so this is per kidney. Alright, so now moving on to the next part. So we will see the structure of a kidney tubule. Okay, so you can see this is the structure, so I'll just draw it and explain. So each kidney tubule is having the following parts. So there are following parts. So there is Bowman's capsule. So before I explain, let me just draw it and explain it to you. Okay, so let's just a second, I'll just change the color. Okay, so now there is kidney. Okay, so this kidney tubule we will see, and also we will see what where exactly arrangement of PCT, DCT, the Henleys loop and all is done. Okay, so now here this one is actually the kidney tubule. Okay, so this is kidney tubule now. So here in the kidney tubule I am just drawing the structure which involves the one sec, so which involves the Bowman's capsule. Okay, so we will see this structure completely. So now this one is a Bowman's capsule. Okay, so this is actually the Bowman's capsule. So this is the one, and then it will continue as such. Okay, so there is a continuation from here. So then this is gonna form the PCT and the DCT. So here is PCT. Okay, so this coiled, we call it as a PCT. Okay, so this coiled structure, what you find, so this is actually the PCT, and you know, so this will continue as such. This goes always reverse. Alright, it's okay. So now we will just draw the Henley's loop. So this part what I have drawn, so this is actually the PCT. One sec. Okay, so this is the part now. So this is actually the Henley's loop. Okay, so this one is Henley's loop. So let me just continue this one. So this one is actually the Henley's loop. Okay, so this is in the form of loop itself. So this is Henley's loop, which will continue as such. Okay, so this is gonna continue as such. So this is how you need to draw this one. Okay, so then it will be joining the collecting duct over here. So here the collecting duct will be there. So let's draw this collecting duct over here. So this is now a collecting duct which is having many openings again. So there are openings over here where other nephrons what are there, so they are also joining. Okay, So this is now a collecting duct. Okay, so this collecting duct, so it will be collecting the urine once the formation of a urine is done. So let's just one part, I'll just erase this part. Oh, this one does not work like this. Okay, anyway, I'll just join it like this then, better. Okay, so this is actually the collecting duct. So now I'll just label the part what I have drawn. So this part what is there, so this is actually the collecting duct, where once the urine is formed, so from different organs, I mean from different nephrons, the urine collection will be done through this collecting duct. Okay, so now here what you find, so this specific structure is actually the Henley's Loop. Okay, so this is Henley's Loop, and here what you find, so this one is PCT. Okay, and this one is DCT. So what is PCT here? So PCT meaning is proximal convoluted tubule. I'll just write it here. So this one is proximal convoluted. Okay, so this is proximal convoluted tubule. Okay, so this is proximal convoluted tubule, and then DCT is there. So okay, so DCT is distal convoluted tubule. Okay, so this one is now distal convoluted tubule. Okay, so this is the structure. Now this part what is there? Okay, so this we can mention, I need to erase this part even though I wrote it, but I need more space for analyzing all this. So maybe I can just erase this part. Okay, I'm really sorry, because I need to mention other parts instead of, so you just know now, right? So what is PCT and what is DCT? So we can just continue with the diagram. All right, so now here in this case, okay, I'll just continue this. Alright, so now here this one is actually the Bowman's capsule. Okay, so this is Bowman's capsule. Okay, so this one is Bowman's capsule, and then we need to see the structure of a glomerulus also. Okay, so I'll just draw the structure of glomerulus, and together we can just make out the different structures what are there. So here what is going to happen, so there will be formation. So there is a arteriole which is coming in. Okay, so this arteriole which is joining here, so this will further divide. Okay, so this is further dividing as such, and it is forming this a mass or a capillary. Okay, so this particular structure what is there, so this we call as a glomerulus. Okay, so this is now a glomerulus, and you need to make sure that, so here the diameter of this tube what is there, so this will be less. Okay, so diameter for this particular efferent arteriole, so that is lesser in comparison with the other one, that is afferent arteriole. So this one is actually the afferent arteriole. Okay, so this is afferent arteriole. Okay, and the other one, so this is what is there, so this is actually the efferent arteriole. Okay, so this is leaving. Okay, so afferent is the one which is coming, efferent is the one which is leaving, so that is why we mention it as a efferent arteriole. Okay, so this is the structure clear? So now this part what is there, so this part we call as a glomerulus. Okay, so this one is glomerulus. So now this Bowman's capsule and together with the glomerulus, so this we call as Malpighian capsule. Okay, so these two parts together. Okay, so these two parts together we call it as a Malpighian capsule. Okay, so this is Malpighian capsule. One second. So I'll just, oh yeah, I was just checking the spelling, so it is correct. Okay, so it is Malpighian capsule. Okay, so this together we can call it as a Malpighian capsule. Is the structure clear? So now we need to see how further division of this particular nephron can occur. So let me just take the different color, maybe not the color actually, No, actually I need an eraser now, so to make this one a clearer diagram maybe. Okay, so just a second, I'll take this. Okay, so here you can see this efferent arteriole, what is that? So this is leaving now. Okay, so this is leaving and it will form a capillary again. So this is the one which is leaving. So this will have a narrow diameter. Okay, narrow diameter as compared to the other one. So what is going to happen? So this will further divide. Okay, so this will further divide, where it is going to form the secondary set of capillaries. So here let's draw the secondary set of capillaries over here. Okay, so secondary set of capillary formation we can see at the PCT. Okay, so now this is a PCT where it is going to further divide. So all of them are again joining back. Okay, so they'll be joining back here, then they are forming this structure, and again there will be division which you will find. So there will be formation of secondary set of capillaries again over here. So again you will find this secondary set of capillaries from here as well. Okay, so this is at this end. So basically we can see this secondary set of capillaries over here. Okay, so now in this structure, so let me just explain this one. So this is further dividing, so we can find further division with respect to this. So this will be further dividing, where it is going to cover the DCT also. So why this is happening, I'll just explain it. Okay, so why this is all happening? So this is mainly happening because all of this, so there are a lot of processes that are going to occur. So that is why there will be formation of secondary set of capillaries which are taking place mainly for absorption, or it can be for the other functions. So it can be with respect to, so mainly the absorption. Okay, so there is selective absorption which occurs. Okay, and here we will see this ultrafiltration process. So here all of these will join together and it is going to join this renal vein. So it will be going to the renal vein. Okay, so now before I explain you the structure, so I'll just explain you something related to this one. So you know heart, right? So heart is there. So from heart, mainly the aorta, what is there? So that is from the left ventricle, the aorta is leaving. Okay, so this is the aorta. Okay, so this aorta which is there, so this is actually the artery only. Okay, so this is artery. So a part of it, it is going to all the different parts. Now the one which is going towards the kidney. Okay, so the one which is going towards the kidney, so let's say here the kidney is there, so it is going towards the kidney. So that is why we call it as a renal artery. Okay, so renal artery is that, so this is the one which is going towards the kidneys, and that is why we call it as a renal artery. So now here the kidney is there. So what will happen? So this renal artery, it has to divide, because you know each kidney, what is there? Okay, so each kidney is having about 1 million nephrons. Okay, so 1 million nephrons are there in each kidney. Okay, so for that what has to happen? So this renal artery has to divide to form the arteriole. So there is a arteriole formation now. So this is one arteriole. So this is actually entering one of the nephron here. So let's say this is a arteriole. So these are arteriole formation. Okay, so this is also again an arteriole over here. So this is also arteriole formation is occurring. So these kidney, that is renal artery, what is them? So renal artery has to divide about 1 million times to form the many different arterioles. So it is to divide 1 million times. That is mainly because here the nephron is there. So this one is a now Bowman's capsule. So what is going to happen? So now this we call as a afferent arteriole. Okay, so this is coming, and here there will be glomerular filtrate, glomerulus occurs. Then there is a efferent arteriole which is further dividing. So same way there will be another nephron. So in one kidney we can find like this millions. Okay, so millions of nephrons are there, and millions of arteriole formation has to occur from the renal artery. So what is going to happen? So this renal artery which will be dividing further, and we can find many arterioles. So this one is again a nephron now, the Bowman's capsule. So this one is actually the Bowman's capsule where the arteriole is further dividing, and there will be formation of efferent arteriole. Okay, so same way there has to be millions of these arterioles which need to be formed as such, and they'll be getting this afferent arteriole. Then there is Bowman's capsule, I mean the glomerulus, and then efferent arteriole. So all these efferent arterioles, what are there? So they'll join together. So let's say now this is the efferent arteriole. Okay, so this is joining. So these are all joining together. Okay, so all these arterioles, what are there? So they'll be joining together, and they'll be joining the renal vein. Okay, so that is basically they'll have most of the carbon dioxide. So this renal vein has to go to the so it will be having deoxygenated blood, right? So it was having the oxygenated blood. Okay, so here the oxygen supply will also be done, waste removal can also be done. Okay, so this deoxygenated blood which is there, so it is going to the vena cava. So inferior vena cava is going to collect this particular blood, that is deoxygenated blood. Then it will be emptied into the right auricle, that is for oxygenation, which will be taken to the lungs later on. Okay, so this is how. So if there is 1 million nephron, then 1 million arterioles are required, and this renal artery has to divide about 1 million times for the formation of, that is to supply the blood to each of this. So you know this renal artery, so this is coming from the renal artery. Okay, so from renal artery what happens? So let's say this is the renal artery now. So this will divide, and it is gonna form this arteriole. Okay, so there is one more division which we can see. So there can be another nephron. So this is forming another afferent arteriole. So there is afferent arteriole, there is efferent arteriole. You can see the diameter over here. Okay, so as in comparison, the diameter is wide. Okay, so wider lumen we can find, and here we will find a narrow lumen. Okay, so now moving on to the next part. Yeah, so other than that, you can see this first convoluted tubule, this I have labeled, then second convoluted tubule I have labeled, then to renal vein I have labeled, the collecting duct is labeled, urine flow we will see. So this one is a Bowman's capsule, this is labeled. Okay, so glomerulus is there, so Bowman's capsule, glomerulus together, we call it as the Malpighian capsule. And this part, okay, so descending limb and ascending limb I haven't mentioned. So this part, what is there? I'll just label it over here. Okay, so this particular part, what is there? So this we call as descending limb. Okay, so this is descending limb, and here since it is moving in this direction, whatever the filtrate which was formed here, so this will be reaching the part over here. So this is the descending limb, and this we can mention as ascending limb of Henley's loop. Okay, so this one is ascending limb. Okay, so here once the urine formation occurs, so urine is going to be collected from different neurons, and this is the urine flow direction. So urine is going to flow in this direction. Okay, so is the structure clear here? So descending limb, and these are the capillary network which I already said. So it is basically a secondary capillary network, and this particular collecting duct, once the urine flow is done, I mean once the urine is formed, so this urine is going to flow, and this collecting duct is going to open up the urine into the pelvis area of kidneys. Okay, so pelvis region of kidney will be receiving this particular urine. Okay, so that is how we are that is how the structure of basically not the structure, arrangement of this particular nephron is done in a kidney. So let's see. Now you can see over here, okay, so this part, okay, so let's say this division if we make over here, okay, so if I just divide this part over here, so this particular part, what is there, so this comes under the cortex region. So under cortex region, what you can find, you will find this dot like appearance. So you can see this kidney structure over here, okay, so this dot like appearance, what is there, so that is mainly because of the what is it, the kidney tubule which is present. So mainly the Bowman's capsule, as well as the, where I had drawn here, yeah, so this is mainly because of the PCT and the Bowman's capsule. So we can also notice a DCT, and this part, what is there, so this comes under medulla region. So medulla region, what is there, so in medulla region we can find the Henley's loop, okay, so Henley's loop, as well as a collecting duct we can find. So this one is actually the collecting duct over here, okay, and on the other side, so in the cortex we can find the Bowman's capsule, then the other one, so basically including Malpighian also we can mention, the PCT, DCT and all can include over here, okay, so I'll just write it PCT over here. So all these are present where? So these are present in the cortex. So we will see the explanation. And why do we call this structure, what is there? Okay. So we call this one as a PCT. Any idea why do we call it as a PCT? Because we call it PCT, meaning is proximal convoluted tubule. Okay. So this one is proximal convoluted tubule, okay, so this is convolutions will be there, so this is actually the PCT, okay, so this is PCT, proximal convoluted tubule, and then there is distal convoluted tubule. So proximal meaning is nearer, okay, so here this is nearer and this is further, okay, so further means far, okay, so this is distal convoluted tubule. So in this case when you observe, so you can see this PCT, proximal convoluted tubule or first convoluted tubule, it is nearer to what? It is nearer to the Bowman's capsule, okay, so nearer to Bowman's capsule, so that is why we have named it as the proximal convoluted tubule, and this one is far from the Bowman's capsule. So you can see the distance over here, so there is, it is far from the Bowman's capsule, that is why further from Bowman's capsule, so that is why the name is kept as or named as distal convoluted tubule. So this is further from Bowman's capsule. Is this clear about the structure? Okay, so now each kidney tubule has the following parts. The Bowman's capsule is there. So it is thin walled single cell thick epithelium cup, okay, so it is actually cup shaped, something like a hollow ball. So you can see this is somewhat cup shaped as such, okay, so here also you can see there is a cup shape, all right, so you can also draw it like that. You need, it need not be in a side angle only, so any angle you can draw it, not a problem, okay, so this is cup shaped and it is having a hollow ball pressed deep down, that is on one side. So you can see this hollow ball like structure. So okay, so this, it's hollow internal space, what is there, it continues into the tubule. Just a second. Yeah, so this continues into the tubule here. So you can see the tubule, the outer concavity of the cup, what is there, so this is going to form a knot like structure, so that is mainly of the blood capillary. So this is all blood capillary, which we call as a glomerulus, and the Bowman's capsule, okay, so now this one is a Bowman's capsule and this particular glomerulus, okay, so Bowman's capsule and the glomerulus together we call it as a Malpighian capsule, or just renal capsule also we can mention. So here Malpighian capsule, or we can also call it as a renal capsule, okay, so now this one is a glomerulus over here and this one is a Bowman's capsule. Next, so proximal or first convoluted tubule, okay, so that is basically PCT. It is the starting convoluted region. One second, I'll just drink some water. Okay. So now the proximal or first convoluted tubule, what is that? So this is a starting convoluted tubule, and that is why the name is proximal. Nearer to what? Nearer to the Bowman's capsule. Both the Bowman's capsule and proximal convoluted part, what are there? So this part, what are there? So this we can find in the cortex region, giving it a dotted appearance in the sectional view. Okay, so then there is the other one, that is middle U-shaped part you can find here. So this is a middle U-shaped part which we call as a loop of Henley. Okay, so this is shaped like a hairpin. So you can find a hairpin like structure. So hairpin bend or hairpin curve when you drive in such cars. So it is basically like this. Okay, so it is not convoluted. Okay, so it is not actually many folds are not there as such. It runs in medulla. Okay, so this is running now. So this particular part, what is this? So it is running into the medullary region, and it is turning back, and it is re-entering the cortex region to continue into the next convoluted region. So you can find DCT also in the cortex region, but the dotted appearance, what is this? So that is mainly in the longitudinal section. So you can see the PCT, but the Bowman's capsule, the DCT is also there in the cortex region itself. Okay, so that is to continue into the next convoluted region of the tubule. So next convoluted region, what is this? So it is distal convoluted tubule. Okay, so now this one is a DCT, again lying in the cortex itself. So it is the end part of the kidney tubule. Okay, so distal means further or farther or away from the Bowman's capsule. So this is gonna open where? So this is gonna open into the collecting duct. So you can see that many nephrons are connected over here. Okay, so through this many nephrons, so basically they'll be emptying their urine into this one. So this is the flow of direction. Okay, basically urine flow direction is this one, where it will be emptying its content into the pelvis, then later on it will be getting into the ureter. Okay, so this will get into the ureter. So here in this case it opens into a collecting duct. The collecting duct is going to receive a contents of many kidney tubules. Okay, so the many kidney tubules will be there. So from everywhere it is going to receive this urine, and it will pour it as urine in the pelvis. Okay, so this is going to pour the secretion or the urine into the pelvis area. Then from pelvis, where it is going, so it will be entering the ureter. Then from ureter it will be entering the urinary bladder. Okay, so this is how the functioning will occur. So uriniferous tubule, we will see this information. So these are tiny. Okay, so uriniferous tubule or nephron or kidney tubule or renal tubule, what we mentioned. So these are all tiny ones. They are so many and for so much. Okay, so they are doing a lot of work. So all the waste which is not required by your body, so this has to be removed out. So what will happen if it remains? So it is very toxic. Okay, so that is why it has to be removed out. And this tiny one which is there, okay, it is removing all the unwanted things. So if it remains in your body, it can be highly toxic, and it can even lead to death of an individual. Okay, so these tiny ones, what are there? The tiny structures or functional units of kidney. So they are doing so much for us that they are removing all the toxic materials from our body. So that that is for so much. Okay, so they are there for so much. So basically total number in both the kidney, approximately we mention it to be 2 millions. Okay, then each single tubule, so now here one tubule is there. So this each single tubule, what is there? So this is one tubule now. Okay, so here is the tubule. So this is somewhat about 4 to 5 centimeter long. Okay, so this will be forming about 4 to 5 centimeter long. Okay, so now if I join, so total length of all the tubules together, so now if I take all of them and if I keep them end to end like this, so it can give us up to 60 kilometers. Okay, so this is gonna form about 60 kilometers if we are taking all these millions together. Nephrons, what are there? So all these millions of nephrons. Okay, so millions of nephrons, what are there? So we'll join end to end. Okay, so join end to end. So in that case, what is going to happen? So this is going to give us about 60 kilometers of them we can find. So this great length, what is there? So it will provide a huge surface area that is mainly for the reabsorption of usable substance. So we will see this reabsorption, ultrafiltration and everything I'll explain. Okay, so that is basically the blood flowing through the kidney per minute is 1 liter. Okay, so 1 liter per minute. So it can flow like about 1 liter of blood can flow through your kidney, that is under 1 minute. Now let's say now in class we are so here YouTube class I'm taking it about 60 minutes. Okay, so basically 60 liters of blood, what is there? So that is getting flowing, or it is flowing. So 60 liters of blood is flowing through this kidney. Okay, so your kidney and my kidney. So basically while I'm sitting over here for 60 minutes, so basically 60 liters of blood is getting filtered. Okay, so glomerular filtrate, so out of this one the glomerular filtrate which is produced in 24 hours, so that is 160 liters. Okay, so from this 160 liters there is absorption that is gonna occur. So urine produced from the glomerular filtrate after reabsorption, so that can be about 120 liters. Okay, so for 24 hours what is happening? So this was for 60 minutes. So let's say for 24 hours how many liters is produced? It is basically 160 liters of glomerular filtrate, not the blood. Okay, so here the transportation they were explaining. Here this one is actually the glomerular filtrate which is produced. Okay, so that is somewhat about 160 liters. So in that, reabsorption will occur, tubular secretion will occur. Okay, so for all those processes, once that occurs, the urine which will be formed, that is per day, so it is approximately 1.2 liter. Okay, so this is how the calculation was done. So now we will see the blood supply to the kidney tubules. Okay, next class I'll explain about the constituents and we can check some question answers also if possible. Okay, 1 second. So today's class anyway I'll explain this. Yeah, so next class we will see this anyway. So I'll just go with this part. All right, so you can see here. Okay, so you need to keep in mind. Okay, so functions of kidney, basically urine formation we are checking here. So pair of renal artery, what are there? So they'll branch off from the dorsal aorta. So this I already said, into the respective kidney. That means you can see here this was a aorta. Okay, so this aorta, what is there? So they have to, this is actually the artery itself, so this has to branch. Okay, so this has to branch, and what is going to happen? It is going to join two of the kidneys. So here one kidney is there and here one kidney is there. So both of these have to receive this renal artery. So once the renal artery, so this is renal artery now. Okay, so this one is also a renal artery. Okay, so from here this is coming. So this renal artery has to divide about 1 million times to form an arteriole. So this is dividing further. So much division is occurring so that it can provide this afferent arteriole to each of these kidney tubules. Okay, so now you can see here, so each renal artery, it is gonna branch, rebranch several times to give rise to arterioles. Okay, so each such arteriole enters the Bowman's capsule. So this we have seen. So this is called as a afferent arteriole. This afferent arteriole is going to break into number of capillaries. So you can see it is further dividing and it is forming the capillary, which forms a knot mass. Okay, so knot like mass, which we call as a glomerulus. So this one is actually the glomerulus, which is fitting inside. Where it is fitting? So it is closely fitting inside the Bowman's capsule. Okay, so now this reuniting, so all these capillaries which were here, so they'll all reunite. Okay, so they'll all reunite and they are going to form this efferent arteriole, that is to go away, it is to carry away. Okay, so this efferent arteriole, after emerging from the Bowman's capsule, so what is going to happen? So it will run a short distance and it is going to break into a secondary capillary network, which we can also mention as a vasa recta. So you can see here, this has come. Okay, so this has come over here. What has done again? So this is broken down again for like further many paths are formed here. Okay, so now this is joining back. So you can see there will be joining back again. This is further dividing. Again there is a formation of the secondary network. So again this will divide over here. So this is again forming this secondary network. Okay, so secondary network or secondary network of capillaries are formed. Then it will go back, I mean all of these will join together. So venule, and this is forming, it is going to join back to the renal vein. Okay, so now by uniting again and again with, just a second. Okay, so now that is by uniting, one second. Yeah, so by uniting and again, so they'll be joining the veins of the kidney, which will ultimately form the renal vein. Okay, so which is going to leave the kidney at the median surface again to pour the blood into the posterior, or which we call as the inferior vena cava. All right, so now we will see the function of this particular kidney. Okay, so function which is performed by the kidney, so that is basically for the formation of urine. So majorly two steps are gonna occur. So formation of urine occurs in two major steps. So that is ultrafiltration, we will see what is it. Okay, then there is reabsorption that is gonna occur. Okay, so now as you can see in this diagram, so you need to make a note here. Okay, so here the diameter is what? The diameter is wider, and here the diameter of this arteriole, what is that? So it is narrow. So what can happen? So as there is a area, or the area available for the blood flow, so you know that whenever volume is gonna decrease, okay, so here the volume is decreased, then in that case the pressure is gonna increase. Okay, so what is happening? So blood is anyway gonna enter through this afferent arteriole. Okay, and here the pressure is built. Okay, so you know all these capillaries are also having a narrow diameter, and with that the efferent arteriole, what is that? So that is also having a narrow diameter. So because of the higher pressure that is built, and that pressure we call as a hydrostatic pressure. Okay, so this is actually the hydrostatic pressure, and because of the hydrostatic pressure, what is happening? So most of the liquid part of the blood, okay, so most of the liquid part of the blood, what is there? Okay, so that is gonna enter into this glomerulus, basically into, so through the glomerulus it is entering into, so liquid part of the blood enters, okay, so this is gonna enter into, okay, so this is gonna enter into the Bowman's capsule. So now this Bowman's capsule has received most of the liquid part of the blood, okay, and this is in case of Bowman's capsule. So now this liquid which is there in the Bowman's capsule, so that we call as glomerular filtrate. Why do we call it as glomerular filtrate? Because it is coming from the glomerulus itself. So because of the narrow diameter, so what is happening, so there is a higher pressure that is built up, and when the higher pressure is built up, what is going to happen, so the pressure is called as a hydrostatic pressure, and most of the liquid part, okay, so most of the liquid part of a blood what is there, okay, so liquid part of the blood what is there, so this is going to enter into the Bowman's capsule, and this particular liquid what is there, so that we call as a glomerular filtrate. Okay, so which is much, so we will just read this one. So the blood flows through the glomerulus under great pressure, which is much greater in the capillaries elsewhere. So the reason for this pressure is basically the efferent arteriole, okay, so it is narrower than the afferent arteriole. So this is narrow, okay, so efferent arteriole what is this, so this is narrow, and the other one, the afferent arteriole, is wider, okay. So now because of that what is happening, so this high pressure, that is hydrostatic pressure, causes the liquid part, okay, so the liquid part of the blood to filter out from the glomerulus, that is into the renal tubule. So this whole process what is there, or this filtration under extraordinary force what is there, okay, so this whole process what is there, so this we call as ultrafiltration, okay, so this is actually the ultrafiltration. So what happens, so during ultrafiltration almost all the liquid part of a blood what is there, basically the plasma along with most of its organic and inorganic substances, including urea, glucose, amino acid, etc. So they are going to come out of this glomerulus, and they'll be passing into this funnel shaped cavity called as a Bowman's capsule. So this one is a funnel shaped cavity, and here all this liquid part of a blood, so basically liquid part when we mention, it can be plasma, okay, so there can be plasma with organic and inorganic substances which can be, again, so it can have urea, then there can be glucose, there can be amino acids, so all these what are there, so they are gonna pass out into this filtrate, okay. So now this fluid entering the renal tubule what is there, so this is actually, so here, yeah, so now the fluid entering the renal tubule, so this filtrate or a liquid is called as a glomerular filtrate. Then this glomerular filtrate what is there, so it will be having the water, it will be having the urea, it will have the salts, it will have the glucose, and other plasma solutes will be there, so it can have all this. So thicker part of the blood, so here since most of the liquid part is entering over here, so the blood which is leaving from this, so the blood will be thicker blood, okay, so the blood which is leaving through this particular ultrafiltration, after the ultrafiltration, so that part will be thicker one, okay. So two kinds of corpuscles, so mainly the thicker part of the blood is left behind, and the two kinds of corpuscles, mainly the proteins and other large molecules, are carried forward. So basically the RBC, WBCs and all will be there, okay, and also the proteins, the larger molecules what are there, so all those will be carried forward through this efferent arteriole itself. So thus the blood which is proceeding away, so this is the blood which is proceeding away from the glomerulus, so this will be relatively thick. So why it will be thick? Because most of the liquid part got filtered over here. So now this all this one, so this is not urine, okay, so this is not urine. So here the reabsorption process has to occur, all the wanted things. So basically selective reabsorption, so all the wanted things what are there, so those need to be absorbed, and then later the ones which is formed, so reabsorption will occur. Later there will be a tubular secretion which we can find, okay. So once that occurs, then only we can call it as a urine, okay. So urine formation we can mention. Maybe I can just show you like this. Maybe I can't move myself there. Oh, you can't see, no? Okay. All right. So this is the one, okay. So after the ultrafiltration process, what will happen is, yeah, so after this ultrafiltration process, we will find this reabsorption process, okay. So next class I'll explain about the reabsorption, so I'll just continue with this one. So I'll just explain you the functions related to ultrafiltration, the reabsorption, and then we will see the components of a urine in the next class, and we will finish this chapter, okay. So before I end the class, let me just say about the certain features that are there, okay. So next class I'll anyway explain about the reabsorption process, okay. So there is Unacademy subscription features, okay. So if you are taking a plus subscription at Unacademy, okay, so you are going to get all these benefits. So in YouTube, all these benefits you are not getting, okay. So you can basically, this one anyway you are there, okay, so it is there. So you can learn live from the comfort of your home. So this will be done. Then there is unlimited access to all the courses. So there are different courses that are going on, okay. So you will have access to different courses if you take the subscription, that is plus subscription on Unacademy. So basically top educators of India are going to take the classes for you. Then there will be regular doubt clearing sessions. So let's say we have conducted about 3 sessions already, and we will be keeping one particular session which is completely for doubt clearing itself, okay. So that we call as a doubt clearing session, and you can clear your doubts in this doubt clearing session, so related to the topics that are covered. Or also we can just, will be giving you question, you can just write down. So basically it can be in the form of poll also, okay. So exhaustive coverage of a syllabus, so complete syllabus will be covered. Then mentorship and guidance is given to you in this one. The study material is given, which is given in the PDF format itself. Then you will be getting the practice tests also. Then there is live test series, you will be getting the batch courses, okay. So there are tests which will be conducted, so you will have access to that as well. Then batch courses, so different batches will be there, so you will have access to different batches also. Then there is daily practice section which will be done. Then there is weekly mock test series, live quizzes, daily MCQ and subjective test series. So all these benefits you can take from the Unacademy subscription. So for that you can use my referral code VANIYT, okay. So this is my referral code VANIYT, where you will be getting 10% discount on your subscription, okay. So you'll be getting 10% off on your subscription, that is for plus subscription. So let's see, so these are the respective months which are given over here, okay. So let's say you are going for any of these months based on your convenience. So all you need to do is enter the educator code, so that you will be getting the 10% discount. So this benefit you can take. So here you can enter the VANIYT, all right. And other than that, so there is this batch which we have recently launched. So this is Udan batch. Basically the delivery of language is Hindi here. So let's say you are taking a subscription for class 10, so you will be getting ICSE class 11 and 12 subscription absolutely free, okay. So here there is a Mavericks batch, okay. So this is again starting from the 17th 19th of January itself. The delivery of language is English here. So let's say you are subscribing for class 10, then you can get ICSE class 11 to 12 subscription absolutely free here as well. Okay, so all these are the same. So this one is already completed. Okay, so you can watch the recordings of this one if you have taken a plus subscription. So basically you can use my code Vani YT. All right, and for this one, this particular batch is also over. Okay, so this one, so again here also you can watch the recordings. Alright, again let's say you are going for a subscription, you can use my referral code Vani YT. Okay, so how you can attend my special classes? So in my special classes, that is on Unacademy Learners app, I'll be giving you polls, I'll be showing you the leaderboards to know your competitors. Then also you can type in your question, you can attach an image and you can send it during a live class, and the educator is gonna clear your doubts during a live class itself. Okay, so what you need to do is like how to watch my special classes. So that is basically all you need to do is download the Unacademy Learners app. Okay, then open this particular app, click on get started. You can also enter your mobile number and OTP. You can sign up using your email ID. Okay, so select your class here. So you can select class 6 to 12. Okay, so for you basically ICSE class 9, 10 you can select. Okay, so you can select your respective class, you can add the goal there as well, and also you can select your preferred language. Okay, so it can be English, Hindi or English. Then go to the description of this particular video and click on the special class link. So you can click on notify me and you will get notified 10 minutes before the class begins. So you can also use my code to unlock the special class for free. Okay, so you can unlock this particular special class for free, that is again by using my referral code Vani YT. Okay, so download the Unacademy Learners app and try all of these features for free for unlocking and for plus subscription. Okay, so for both of this you can use my referral code Vani YT, where you will be getting, so for plus you'll be getting 10% off. Okay, so this is Vani YT. So see you all in the next section. Okay, so next session is actually on this function related to uriniferous tubule itself. Okay, so that is functional unit of a kidney we will see, and after that I'll be just covering some urine constituents and certain information related to our disorder conditions of this one. Okay, so see you all in the next session. Okay, bye bye everyone.